Audio Jungle. Welcome back to another video tutorial on acid fast staining. Okay, acid fast stain. So yes, so this is another method of differential staining technique. Okay, so differential staining technique means, yes, here also we differentiate between two groups of organisms, that is mainly acid fast group or acid fast bacteria with non-acid fast group of bacteria. Okay, so so this staining technique is also known by the name which is called as Zeal-Nielsen staining. Okay, so here Zeal and Nielsen are two separate persons, two different persons, where both have contributed for this technique. Okay, and Zeal is the one who introduces his carbalfuxin, who uses carbalfuxin for this technique, and so Nielsen is the one who gives you a correct method of performing this staining technique, and hence we call it as Zeal-Nielsen staining, as both of them has contributed to the technique. Okay, see. Nielsen in 1883 used Zeal's carbalfuxin and heat and then decolorized with an acid alcohol and counterstained with methylene blue. So this technique was given by him by using Zeal's carbalfuxin. Okay, and yes, as I told, so this method is mainly used to differentiate between acid fast bacteria and non-acid fast bacteria. Okay, and what is this acid fast bacteria? So non-acid fast bacteria. See, now acid fast bacteria is mainly Mycobacterium, especially Mycobacterium leprae, Mycobacterium tuberculosis, and Nocardia. Okay, why do we call them as acid fast? Is because these acid fast bacteria, no, they have a special coating which is called as mycolic acid layer surrounding their cell wall. Okay, and so this will make the cell wall very waxy, and because of this, no, so the stains cannot enter inside the cell very easily, and we cannot use the simple staining techniques like gram staining over here. Okay, so and hence we use a special stain which is called as carbalfuxin, which contains phenolic compounds in it. Okay, and we also use a special technique to make the cells penetrate inside such bacteria. Okay, and once when the bacteria is stained, it cannot get decolorized very easily, and the stain will not come out very easily, and hence we call it as acid fast bacteria. Okay, it is difficult for it to get stained, and once it is stained, so it is not easy, or it will not get decolorized at all. Hence we call it as acid fast. It retains the color. Okay, so hence we use a special technique here, special stain here, to forcefully introduce the stain, and it will not even get decolorized. Okay, so this method is acid fast staining. Now, so next, so is a principle of acid fast stain. So acid fast mycobacteria contains mycolic acid in their outer membrane, making the cell waxy and resistant to staining. With aqueous based stains such as gram stain, the primary stain carbalfuxin is applied to the cells, and heat and phenol are used to allow the stain to penetrate into the waxy surface of the acid fast microorganism. The excess stain is removed with the treatment by acid alcohol. A secondary stain methylene blue is then applied to the cells. Okay, now as you can see in the picture over here, so this is a waxy layer or the mycolic acid layer which is present in the mycobacterial cell wall. Okay, and so this mycolic acid is the one which is very tough. They do not allow the stain to enter inside very easily. Okay, and because this mycolic acid layer is present now, so you cannot stain them with normal aqueous based stains such as gram stain, and you require a special stain like carbalfuxin. As I told, carbalfuxin will contain phenolic compounds, so this phenolic compounds will solubilize this mycolic acid layer. So since it solubilizes, no, so it will allow the carbalfuxin to move inside the cell. And apart from this, you also apply heat over here. Okay, so just by pouring the stain onto the smear, no, so it will not absorb so much. So you need to heat them also. Okay, so you will gently place the slide on the water bath, and you will keep on heating the slide, and you will keep on adding the carbamazepine on top of it. So this particular heating method also will be done for about 3 to 5 minutes. It will continuously be done, so that forcefully you are introducing the carbamazepine into the cell. So and once you have introduced the carbamazepine, then if you try to decolorize, what happens? It will not get decolorized. And if it is non acid fast bacteria, so easily it will get decolorized because there is no mycolic acid layer present. Okay, it will get decolorized and it will appear colorless. And finally, when we add methylene blue, that is a counter stain, so this appears red itself because it is not decolorized, red is still present and it appears red only, acid fast bacteria. Whereas if it is non-acid fast bacteria, so at that now it is colorless and it will take the counter stain methylene blue and that appears blue in color. So this is a basic principle of acid fast stain. Now see, when the smear is stained with carbamazepine, it solubilizes the lipodial material present in the bacterial cell wall, but by the application of heat, carbamazepine further penetrates through lipodial wall and enters into the cytoplasm. This is what happens in the first step. Okay, first you will take a slide, you will make a smear. So after making the smear, no, so you will put the carbamazepine on top of it. So this carbamazepine is made up of phenol, and as a result it solubilizes the lipodial material, that is mycolic acid. It solubilizes the mycolic acid layer in the cell wall, and further you will apply heat, and this heat also will help in the carbamazepine to move inside the cytoplasm. Okay, now with the help of heat and with the help of a special stain, you have stained the mycobacterium. And as I told, once you have stained, it is difficult to remove. Then after the cell appears red, now the cell is red. Then the smear is decolorized with decolorizing agent, but the acid fast cells are resistant due to the presence of large amount of lipodial material in their cell wall, which prevents the penetration of the decolorizing solution. So in the second step, when you add decolorizer, what happens? In the acid fast bacteria, so the decolorization will not happen because the decolorizer cannot enter inside the bacterial cell because it's made up of high lipodial material. Whereas if it is non-acid fast bacteria, what happens? It will get decolorized very easily. And in the last step, what happens is the non-acid fast organisms lack the lipodial material in the cell wall, due to which they are easily decolorized, leaving the cells colorless. So when the smear is stained with counter stain methylene blue, only decolorized cells absorb the counter stain and takes its color and appears blue, while the acid fast cells retain the red color. Okay. So what happens now? So if it is acid fast, so it cannot get decolorized, and if it is non-acid fast, it will get decolorized. And in the last step, when you add the counter stain, that is methylene blue, acid fast bacterium will not take that because it is not even decolorized and it appears red itself. Whereas if it is non-acid fast, so it takes up the methylene blue stain because it's already decolorized and it appears blue in color. Okay, so that's what happens over here. And then this is a principle of um, acid fast staining. Okay, let's move on to the procedure over here. See, first prepare and fix the specimen smear prior to staining. Yes, you will take a clean glass slide. Okay, you will prepare a smear of sputum or whatever sample you have, and then you will air dry that, heat fix that. After that, place a small strip of blotting or filter paper over the top of the specimen and place the slide over the boiling hot water bath on a mesh surface. Cover the filter paper with a primary stain carbamazepine. Leave the slide on the water bath for 3 to 5 minutes. Continue to apply stain if the filter paper begins to dry. Yeah, so this is a special technique which we follow for the carbamazepine to enter inside the cytoplasm of the cell. See here, first we will take a glass slide. On top of it there is a smear. On top of it you will put a blotting paper or filter paper, and this slide itself you will go keep it on the water bath. Okay, and you will keep on adding carbamazepine once the blotting paper absorbs and it gets dried. You'll keep on adding and you'll keep on steaming for about 3 to 5 minutes. So this is made for the carbamazepine to penetrate into the bacterial cytoplasm, and this method or this procedure will make the carbamazepine enter into the cell. Okay. So yes, and then remove the filter paper and rinse the slide with water until the solution runs clear. So once this step is done, now you will wash it, you will water wash it to remove the excess stain. And then after this, what happens? Run the acid alcohol decolorizer over the slide for approximately 10 to 15 seconds. So now you will take the decolorizer, okay, and you will add on top of it, and then you will allow it to remain for 10 to 15 seconds. So at this point, what happens? If it is acid fast bacteria will not get decolorized and the color will remain red only. Whereas if it is non-acid fast, mycolic acid will not be present, and as a result easily it will get decolorized and it will appear colorless at this stage. Okay, and then next is you will add methylene blue, and then you will air dry and you will observe under microscope. See. Rinse the slide with water. Cover the smear with a secondary or counter stain methylene blue for 1 minute. Gently rinse the slide with water. Blot the slide air dry with fibulous paper. Okay, so next, in the next step, you will again water wash, and then you will add the methylene blue. So at this step, when you add methylene blue, what happens? So the acid fast bacteria cannot take, and it remains red as it is. Whereas if it is non-acid fast, it is decolorized, it's already colorless now, and as a result it will appear blue in color. Okay, so then again you can rinse with water to remove the excess stain, and then you can blot dry and you can observe it under microscope. So what you can see when you observe under microscope, bright red to intense purple, red straight or slightly curved rods occurring singly or in small groups may appear beaded. So blue color, in addition, background material should stain blue. Okay, that means simply if I want to tell, the red colored things which you can see is acid fast bacteria. Okay, and the non-acid fast bacteria will appear blue in color. Okay, see if it is acid fast, it is bright red to intense purple in color. Red color it will appear. If it is non-acid fast, it will appear blue in color. So you can identify based on the color over here. See. So this is the steps over here which is given. See, first when you apply the counter, that is, sorry, primary stain, so all the primary stain, and when you heat, no, all the cells over here have taken up the primary stain and it has appeared red in color. Okay, see, you're heating, and at this particular stage all are red in color. Then after this, when you're adding decolorizer, what happens? So this red color ones are acid fast bacteria and they remain as it is because they don't get decolorized. Whereas other bacteria which is non-acid fast, no, they get decolorized and they appear colorless. Okay, so then when you add methylene blue, what happens? So this red color acid fast bacteria will remain as it is, whereas the non-acid fast bacteria will appear blue in color. So when you observe under microscope, you can differentiate easily based on the color differentiation.